Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic cross in Drosophila, and today we are going to focus on 50% female lethality in Drosophila regarding Carli mutation. Okay. So in this series of genetic cross in Drosophila, I have already made 6 videos. The first 3 videos are on eye color mutation, vestigial wing mutation and Carli mutation and related crosses in Drosophila. In the 4th, 5th and 6th video, we have discussed about male lethality in Drosophila. The first video, in the 4th and 5th video, I have discussed about 50% and 100% male lethality, and in the 6th video, I have discussed about triple lethality in Drosophila regarding Carli mutation. So if you want to watch those videos, the link will be given in the description box below and also in the I button, and I will also attach the playlist link at the end of this video. You can check it out. Okay, so without further delay, let's begin. And before beginning, I would like to request you to kindly subscribe to my channel for more such videos, and if you like this video, if you find it useful, don't forget to hit the like button. So okay, let's begin. See here, first of all, what is Carli mutation? In my previous videos, I have already discussed in detail what is Carli mutation. So here I am just briefing it. Carli is a mutant allele denoted by CY. Okay. But you have to remember that though it is a mutant allele, but phenotypically it is dominant over wild type of wing allele, that is denoted as CY plus. Okay, so CY is dominant over CY plus phenotypically. Okay, and as the alleles are present on the second chromosome of Drosophila, so it is known as autosomal dominant allele. Carli is here autosomal dominant allele. So obviously, as Carli is a autosomal dominant allele, in homozygous and heterozygous both condition, it should show Carli phenotype. Okay, so you can see here in heterozygous condition CY plus is showing Carli wing. But in homozygous condition CY is showing lethal. Okay, that means the progeny is not survived. Okay, is not surviving. So what is the reason behind that? The reason is that Carli is CY mutant allele is also a recessive lethal allele. Okay, though phenotypically it is dominant over wild type wing allele, but it is also a recessive lethal allele. That is why in homozygous condition it shows lethality. Okay, the progeny does not survive. And as we know that CY plus is recessive to the CY, so obviously in homozygous recessive condition CY plus will show wild type of wing. Okay, in wild type of wing we can see that Drosophila has a normal wing like this, but in case of Carli phenotype we can see that the wing is curled backwardly. Okay, got it? So this is all about Carli mutation. Now what is 50% female lethality? See, 50% female lethality means 50% of the female progeny of the F1 generation is dead here. Okay, so to kill the 50% female in the F1 generation, we have to take a lethal allele known as L on the X chromosome. Okay, and you must remember that L plus is dominant over L. So L is the lethal mutant allele and L plus is the wild type allele. Okay, so if there is L plus or L plus L, the progeny survives, but if there is LL, okay, then the progeny gets dead. Okay, so let's understand this with an example or with a problem. See here, the problem asks, this is the F1 progeny data given here. You can see here there are 20 Carli male, 10 wild male, 10 Carli female and 5 wild male. So you have to generate the P generation cross from this data. Okay, these are the F1 generation progeny data. So first of all, we have to derive the sex ratio. The sex ratio is male is to female equal to how many males are there? 20 plus 10, 30 males are there, and how many females? 10 plus 5, 15 females. So it is 2:1. So male is to female ratio equal to 2:1. That means 50% female progeny are dead here. Oh, okay. So we have to take a lethal allele L to kill the females here. Okay. Now phenotypic ratio, that is Carli is to wild, equal to how many Carli phenotype Drosophila are here? 20 plus 10, 30. Okay, and how many wild types? 10 plus 5, 15. Okay, so it is also 2:1, and we know that when recessive lethal allele is present, the monohybrid cross ratio becomes modified from 3:1 to 2:1. As here the Carli mutant allele is a recessive lethal allele, so it is a monohybrid cross ratio. Okay. In my previous videos, I have taught you that whenever you will see monohybrid cross ratio, then we must take both the parents in the parental generation as heterozygotes. Okay, so see here the female is taken as heterozygote having CY and CY plus, and male is, I have not written male here, it is the male parent, it is also taken as heterozygote CY, CY plus. Okay, now these CY are in the autosome or second chromosome. Okay, now how will you design the X chromosome or X and Y chromosome in this case? See here, in case of 50% male lethality, we have seen that the female must have L plus and L alleles on each of their X chromosome. Okay, one X chromosome of female contains L plus allele and another X chromosome contains L allele. Okay, now in that condition we took male as L plus, okay, on its X chromosome, and a Y allele and the Y chromosome as it is. Okay, so that when these this X chromosome of female will cross with this Y chromosome, the male will survive. Okay, but when this X chromosome of the female will cross with Y chromosome of the male, that male will get dead. Okay, so 50%, that is how 50% males are being killed in the F1 generation. Okay, but here the condition is different. Here you have to kill 50% female. Okay, so instead of taking this thing in the X and Y chromosome of the father, we have to delete the L plus portion of the gene or that loci from the male, and we have to translocate that on the Y chromosome. Okay, you can see here we have translocated L plus from here to here on the X chromosome. Okay, so translocation of L plus allele from X chromosome to Y chromosome in males. Okay, that means this part that is containing L plus is cut and then it was translocated to the Y chromosome. Got it? So now this area is divided of L plus allele. Okay, got it? Now what will happen? See here, in this case, whenever this L plus will react with this Y chromosome, okay, this X chromosome will react with this Y chromosome, the male progeny will survive. Okay, because both of them have L plus allele. And when this L will combine with this Y chromosome, it will also survive. This male progeny will also survive because it has L plus allele on the Y chromosome. Okay, which complements the absence of L plus on the X chromosome from mother. Okay, got it. Now let's take another color from here so that you can easily understand. See here, in case of female progenies, what will happen? If this L plus is crossed with, is combined with this X chromosome from the father, this female progeny will survive. Okay, why it will survive? Because though this X chromosome of the father is lacking L plus, but mother has L plus, so this one will survive. We know that L plus is dominant over L, so it will survive. But see, when this X chromosome of mother will combine with this X chromosome of father, this female progeny will get dead. Why? Because in this X chromosome of the mother, L is there. Okay, and to complement that, you need a L plus from father. But in this X chromosome of father, there is no L plus, so obviously this female progeny will get dead. Okay, so you can see here both the male progenies are surviving, but only the 50% female progeny is surviving and 50% is being dead. Okay, so that is why we have generated this type of mutants in the parental generation, so that in the F1 generation 100% male progenies are survived, but only 50% females get survived. Okay, so let's do the cross from this. Okay, I hope this is easily understandable to you. Okay, if you have any doubt, you can leave a comment below in the comment section. I will clear the doubt. Okay, so see how many gametes can be formed from this. So from here 4 types of gametes can be formed from mother. One will be CY with the X chromosome having L plus, then another one will be CY with the X chromosome having L allele. Okay, then CY plus with L plus. Okay, CY plus with L plus, and another will be CY with L, CY with L. Okay, from the father one will be CY with this X chromosome. Okay, then CY with the this new Y chromosome. Okay, then another one will be CY plus with this X chromosome, and then CY plus with this Y chromosome. Okay, so these 4 gametes will be formed from the father. Now putting all these gametes in the checker box, okay, we can see that. Let me just, okay, we have put the male gametes over here and female gametes over here, and you can see how we will determine the phenotype of the progenies here in the F1 generation. See here, in here you can see CY and 2 X chromosome, but see, as we know that CY is a recessive lethal allele, so whenever CY is present, it will get dead, so it is a lethal female. Okay, then similarly it is also a lethal female due to presence of CY here. Then CY plus with 2 X chromosome, one having L plus, so it will be a surviving female, curly female. Then CY plus, so it is a curly female, but as it lacks the L plus allele, okay, no L plus allele is here, so it will be a lethal female. Okay, then CY due to presence of CY, it is already dead. Okay, here also you can see CY is present, so this is a lethal male. Okay, due to presence of CY. Then CY plus, so it is curly, and it has X and Y chromosome with L plus allele, so it is a surviving curly male. Then CY plus, so it is a male with L and L plus allele, so it is a surviving curly male. Then CY plus CY, so it is curly, and here 2 X chromosomes are there, one with L plus, so it is a surviving curly female. Then CY plus CY with only a single L allele, no L plus allele is there, so it will be lethal due to absence of L plus allele here. Okay, then CY plus CY, so it is wild type, and due to presence of L plus allele on one of the X chromosome, it is a wild type female. Then CY plus CY, so it should be a wild type female, but as there is absence of L plus allele in here, so it is lethal female. Okay, got it? So due to absence of L plus, it is a lethal female. Then CY plus CY, so it is curly, and X and Y both having L plus allele, so it is a curly male. Then CY plus CY, curly, X and Y chromosome, one have L plus allele, so it is a surviving curly male. CY plus CY, so it is wild type, X and Y chromosome both having L plus allele, it is a surviving wild male. CY plus CY, it is wild type with one with a Y chromosome having L plus allele, so it is a surviving wild male. Okay, so from this you can see here, from the 16 progenies that could have formed, 1, 2, 3, 4, 5, 6 and 7 are dead, okay, or lethal. So among 16 progenies that could have formed, 7 are lethal and 9 is surviving. And among these 9 surviving individuals, we can see that how many curly male, curly female and wild females are there. Let's count. How many curly males are there? Let's count how many curly males are there. See here, this is one curly male, then this is one curly male, 7, 8, then 13 and 14. So 4 curly males are there. 4 curly males are there with their respective box number 7, 8, 13, 14. Got it? Then how many wild males are there? See, there are only 2 wild males, the box number 15 and 16. Okay, so there are only 2 wild males. Then how many curly females are there? We can see there are only 2 curly females. One is here in the box number 3 and another one is here in the box number 9. Okay, so there is only 2 curly females in box number 3 and 9. Okay, and how many wild females are there? These are the 2 curly females, and how many wild females are there? There is only a single wild female, that is in the box number 11. Okay, in the box number 11. So 4 plus 2 6 plus 2 8 1 9. So 9 surviving individuals are in this ratio. Okay, so from the question we can see what is our given ratio. This is our given ratio. So we can see here 1 is 2, 2 is 2, 2 is to 4. This is the ratio. Okay, so the given ratio is, see here, the given ratio is curly male is to wild male is to curly female is to wild female equal to 20 is to 10 is to 10 is to 5. So 4 is to 2 is to 2 is to 1. And from our cross we have got the same ratio, that is, see here, that is from the observed in our cross we have observed the same ratio, that is 4 is to 2 is to 2 is to 1. Okay, 4 is to 2 is to 2 is to 1. So the ratio are similar, same. So our cross is correct and it is proved. Okay. Now among these 7 lethal, see there are only 1 lethal male. Okay. So where is the lethal male? This is the lethal male in the box number 6. Okay, so box number 6 carries only 1 lethal male due to CY. Okay, recessive lethal allele CY, and there are 6 lethal females. Okay, among these 6 lethal females we can see 3 females are dead due to presence of CY recessive lethal alleles, and 3 females are dead due to absence of L plus allele in their X chromosome. Okay, so 3 females which are dead due to CY alleles are in the box number 1, 2 and 5. Let's see where are those. See here in box number 1, 2 and 5. These 3 females are dead due to CY alleles present in recessive homozygous recessive condition. And in box 4, 10 and 12, these females are dead due to absence of L plus allele. Okay, so see, let's see box 4, 10 and 12. See here in the box 4, you can see there is absence of L plus allele. So this is dead due to absence of L plus allele. If L plus allele was present, it could be a curly female. Okay, then box 10, see here, it is also dead due to absence of L plus allele. If there was L plus allele present, it could be a curly female. Okay, and you can see the box 12 here, it could be a wild female as it has CY plus, but as it has no L plus allele, it is lethal. Okay, so these 3 are dead due to absence of L plus allele, and 4th and 10th box indicates that if they survived, they could be curly, and the 12th box designates that if they survived, they could be wild type. Okay, so this is the calculation of 7 lethal alleles, 7 lethal progenies here, and this is the calculation of 9 surviving individuals from this cross. Okay, so I hope the cross is clear to your mind now. Okay, how to design a cross for 50% female lethality in curly mutation, you can do it now. Okay, so if this video is useful to you, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.